I'm a practicing transplant and leukemia clinician in Birmingham, UK, right at the heart of the UK, and we have one of the biggest catchment regions in Europe. And I've spent the last 25 years trying to develop strategies that accelerate patients' access to clinical trials. So we're just going to talk a little bit through clinical trials and then perhaps open up to some questions. So next slide, please, Natasha. So I think the most important underpinning concept here is that really ever since Watson and Crick understood the structure of DNA, there has been investment of billions, probably trillions of pounds, euros, tax dollars into basic biomedical science predicated on the fact that if we understand the biology of cancers, many diseases, but in this case, leukemia, we will be able to improve outcomes for patients by developing new treatments. And really until about really probably 1999, when Brian Drucker presented the American Society of Hematology on a rainy Saturday in New Orleans, the extraordinary data in which he showed that by understanding the molecular basis of chronic myeloid leukemia, in other words, that there was a translocation chromosomes 922, the Philadelphia chromosome, generating a new gene, BCR-ABL, which constitutively drove proliferation of cells. He was able to work with John Goldman and Novartis to develop a tablet taken once a day that blocked the activity of that activating mutation. And patients with chronic myeloid leukemia, as we know, now have outstanding responses to imatinib and second generation drugs. But the progress actually in terms of translating our understanding of the molecular basis of acute myeloid leukemia to new treatments has been much slower. But in the last 10 years, there has really been hope and a change in clinical outcomes for particular populations of patients. I think we'd probably say older patients who benefit from venetoclax, and patients with particular mutations, such as a FLT3 mutation or an IV mutation. So we're now in a situation where the pharmaceutical companies across the world are using the information generated by brilliant scientists to develop new drugs. And often, as we all know, these drugs can be powerfully effective for patients, either prolonging remission or actually in some patients curing them. And so I think it's really important to have that societal context, which is that everybody on this call and their parents and their grandparents have invested their money into the development of these treatments. And for 50 years, the dividend was deferred. There really was very little in terms of new treatments until recently. But now we have this range of new drugs that's only been developed because of your investment into basic science. And what we have to do, therefore, almost as a moral necessity, is ensure that patients have knowledge about clinical trials of new treatments, have access to these new treatments through trials, and that we move, therefore, to a process of accelerated approval of the drugs that are safe and effective. And that actually means reconfiguring how we spend medical research money. It is now vital that we connect patients at scale with new drugs through clinical trials, and that doesn't always happen. How do we do clinical trials? Well, typically there are a number of stages. The first would be to take a new drug and look at the toxicity of the safety of that drug. And that also, that phase 1 trial is very important in what we call determining the maximum tolerated dose. We then move on with a drug that is tolerated and at a sensible dose to a phase 2 study. This could be a single arm phase 2 study where we look at further signals of efficacy, or it could be a randomized phase 2 study where we look at the drug versus a placebo control in a smaller population. And both of those can be used to say, well, not only is this drug well tolerated, has an acceptable safety profile, but it seems to have promising signs of activity. And then a phase 3 trial is a larger study which provides definitive evidence, usually in populations between 300 and 500, to show that this drug, either alone or in combination, is better than the standard of care. And then that leads to approval by either the FDA or the European Medicine Agency, and the drug then can be widely prescribed, is licensed, and gets its way into patients. Then once that's happened, we also like to see whether the drug is as good as the clinical trial suggested it was. And we'll come on to that little piece in a minute. So that's the phasing of clinical trials. Next slide, please. I think I've touched on some of the questions. The two pivotal questions. First, most important question is this drug safe? Remember the thalidomide scandal in the 60s and 70s. We simply cannot have a situation where a drug is prescribed, whether it's in leukemia or any disease, which has an unacceptable safety. So that's very important. The second question is measures of activity. Does it benefit patients? And there are a number of ways that we can assess that. We can look at, does it prolong survival? Does it increase the remission rate? Does it prevent relapse? And importantly, increasingly, people are also saying, does it improve the quality of life of patients? We're using measures called PROMs, patient-reported outcome measures. So a clinical trial in the early phase is focusing on assessment of toxicity and safety and early signals of activity. And then when we get onto the phase 2 and phase 3 studies, they're really focused on, does this drug look like it's moving the bar for patient benefit? Next slide, please. There is a complex, correctly, process for governance of clinical trials. I'm not going to go through this in detail, but there are some very important considerations. In America, the FDA have to approve the design of the trial. In Europe, it's a bit freer. Second is you have to get ethical approval. A panel of people who may have patient representatives, but from a broader perspective, to check that the questions that are being asked in this study are not only valid, but they're fair for patients. Particularly, is the control arm, is that the appropriate control arm? Is the risk to the patient mitigated? Is it an acceptable risk? And once the trial has gone through the regulatory process, the trial is opened for phase 1-2 trials where you only need about 25 or 30 patients. It may only be opened at 2 or 3 sites. But for randomized phase 2 or phase 3, you want to recruit as quickly as possible. So you may open at 15, 20, 25, or for an international phase 3, it may have something like 80 sites across the most important step then is that something called a patient information sheet is given to the patient. And this, after it's been introduced by the clinician or the nurse, is a summary for the patient. What's the purpose of the trial? What does the trial look like? And what are the possible risks? I mean, I would say to our research nurses that the only time the patients I see in my clinic, actually I see them reading papyrus or paper, is when they get a patient information sheet. The rest of the time, particularly the younger patients, they're on their phones or computers. And I think we are actually very much behind the curve in terms of how we deliver patient information to patients in a timely and appropriate way. And actually for the UK transplant studies in AML, we've developed, I'm sure other groups have, very nice little videos on YouTube, which are much more accessible. So there's a process whereby patients are informed about the structure of the study. They then have a chance to think about it, come back to see their clinician. And if they're eligible, this is really important, is do they meet certain criteria in terms usually of fitness, one sort of disease, then they can be consented to participate in the trial. It's very important during that process that patients are informed it's perfectly reasonable for them not to go onto a trial. It won't affect their treatment. Or if they are on the trial, they can always withdraw. Then the patient is screened to check that, yes, they do meet these criteria. They're renal function's right, their liver function's right, they certainly do have the sort of leukemia. And then they would have a trial visit to assess baseline features. These are mostly lab tests, and see for women, it's very important to check they're not pregnant. And then they would be randomized through a process that we have no control over, typically to the treatment or the control arm. Patients often say, well, what happens if I get the control arm, doc? What's the point of going on to a clinical trial where I get the control arm? And I think one of the most strikingly important features of clinical trials is the data in all diseases, not just leukemia, that patients who are treated in a randomized trial on the control arm, the outcome on the control arm is always better than you anticipate and better than historical controls. It's a really interesting fact. That's true in diabetes, true in autoimmune disease, it's true in leukemia. The randomized trials I've done, the control arm always did better than you think. That may be because of patient selection, maybe it's because of improved standards of care, or maybe because of an extra layer of scrutiny that you get by being on the study. So it's important to assure patients they're very unlikely to come to harm. And even if they're on the control arm, there may be a benefit. And for many patients, also the notion of altruistically contributing to improving outcomes for the general population is powerful. When patients receive the treatment as per the outline, the response is measured, assessment of any toxicities measured. That's completed typically now what we call an ECRF, an electronic form. Come to the end of the trial, the period of treatment stops, and then there's usually a period of 1 or 2 years for follow-up. And in a randomized study, we're looking at simple, straightforward measures. What was the safety? Was there any toxicity signal between the control and the experimental arm? What was the remission rate? What was what we call the event-free survival? In other words, how long people were alive without any evidence of disease coming back, and what was overall survival. We have those data, and then it's important that we accelerate the analysis of that data and publication and dissemination through, first of all, abstracts presented at international meetings, and then a definitive peer-reviewed publication. It's really important that's done quickly. So particularly if it's a positive study, patients across the world can benefit as quickly as possible. There's no excuse for sitting on this data. And equally, if it's a negative study, that may also be very important in terms of stopping particular treatments. I was a chief investigator on a study called Figure, a transplant study, where we looked at in older patients having a transplant for AML, an intensified treatment regimen. It was really quite inconvenient for patients. We did the study because the early phase studies looked promising. There was no difference. So we were really keen to publish that data so that regimen isn't used. So the extra week in hospital patients had to have because of that trial, we were able to get rid of that and actually saved, of course, patients a lot of time and the health service a lot of money. So next slide, please. And I think it's really important that we're empowering patients so that they have the information about relevant trials, that there's access to websites that patients, particularly at diagnosis or disease recurrence, can ask their consultant if there are any available trials, but there's also websites, particularly clinicaltrials.gov, which can give you these data. Next slide. So I suppose these are the key points, really. I think it's the responsibility of doctors and nurses to tell patients that there are trials available, and this should be a default option. But it's important also for patients to be empowered to ask if there's a trial. The patient and the doctor can then discuss this. You can also, of course, encourage your clinician to reach out and see if there are any trials available, maybe not at your hospital, but another hospital. And there are also registries of volunteers. Next slide. So I think I've made these points. It's very important that patients are helping us to design clinical trials. Particularly in the UK, we're using PPIE a lot, patient and public engagement. And actually, the evidence is that when patients help design a trial, the trial's always more effective. And we also need to be thinking very much as a community about how do we make sure that there's enough resource in the hospital in terms of research nurses and clinicians' time so that all patients can be offered trials when they're available. Often patients aren't offered a trial simply because there isn't the resource. And we therefore need to be growing research nurse skill sets. We need to grow national networks so that patients can, as a default, have access to a clinical trial. And it's really important because it allows us to assess these new drugs as quickly as possible. Next slide. And this is final information. So just a couple of closing remarks, really. I think the first is to emphasize that the reason we have new drugs is because of the investment of patients and their relatives over the years. And it's now time to recoup that benefit. We must make it as easy as possible for patients to access drug transplant trials. One. Secondly, it's very important to recognize that there are roadblocks in terms of recruiting patients to trials. We should be investing much more money into clinical trial networks with empowered research nurses so that we can recruit patients to clinical trials when patients want them. So we accelerate the assessment of these new treatments. Finally, you've probably heard about registries, real-world data. This is a growing area of interest. Many patients don't meet the criteria for a clinical trial. That may be on the basis of their kidney function, liver function, age, comorbidity. And registry data collecting how the patients treated in the real world is increasingly important in allowing us to evaluate the best treatment options for those patients who didn't have a criteria to go into the pivotal trial. So I think you'll hear a lot more about registry as real-world data, which we should see as complementary to a mixed, broad portfolio of accelerated clinical trials of patients with AML. And it's through that that we're going to steadily improve outcomes in patients and establish this breaking wave, exciting breaking wave of new therapies as standard of care for patients with AML. Very happy to take any questions.
I think you talk about the national registries, right, and empowering patients to find clinical trials. And so I loved seeing that you had our LLS clinical trials on your resources because we have a very user-friendly system where we have a clinical trial support center at LLS. So we have clinical trial nurses who will search for patients given all of their treatments that they can tell us. They go through and handpick for inclusion, exclusion criteria and compile potential clinical trials, like true potential clinical trials, not like what you find if you like do a web search, that they can take back to their treatment teams to say, hey, okay, are any of these an option or do any of these look better than the standard of care for me? And then they can help navigate that if the hospital staff doesn't have the capacity to do that. So we do that for the US and Canada, so North America. It's a resource that I can't tell you how many patients, once they learn about it, if they're looking for clinical trials, it's wonderful. And even if they don't find a trial, they at least have gotten educated about trials and they know they can come back to it in the future. And they also know that they've looked at all their options. So they feel like they're educated on and making treatment decisions. So that's my just soapbox for that national registry.
That's fantastic. Fantastic. And the other thing I think LLS has done, which is so brilliant, is the therapy acceleration program, which inspired the UK Trials Acceleration Program, putting research nurses into networks of hospitals so we can rapidly recruit patients to pivotal trials. It sometimes takes 5 years to recruit patients to pivotal studies. That's much too long. And we need to be investing, as LLS has done, and our charity, Cure Leukemia in the UK, we need to be investing major amounts of money into ensuring that we can deliver these trials to the highest possible standard to as many patients as possible. So I think we're talking about twin propositions, but your registry sounds amazing. Thank you for continuing to inspire us.
Thank you.
I have a question and a comment, Dr. Craddock. Thank you very much for that. And Stacy, the LLS has been such a supportive thing for me. But when I listened to Dr. Craddock talking about the complexity of clinical trials, talking about 5 years. And I was just wondering, it occurred to me, in the United States, for people who are terminally ill, there's this option of doing clinical trials called Why Not Try? So my question is, and I don't expect an answer, but what about, and you may not have been on when I said when I did my clinical trial, I was told it's like 3% of people would take a trial as their first option. So what about the concept of something between a standard of care and a clinical trial for something that maybe, you know, almost there, almost pretty good. So I'm just throwing that up as the clinical trial, that those two words cause it to have to go through every government, every review, and it's such a complex thing. So might we consider getting something between a standard of care and a clinical trial treatment quicker to patients?
Yeah, I couldn't agree with you more. I think the first observation is that the process for approving trials is excessively bureaucratic. A lot of the questions that patients have to fill, a lot of the hoops we have to jump through aren't necessary. And when you speak to patients, they say, please, can you just speed this whole thing up? Okay. So I think we should be challenging government regulations for fit for purpose. The second is, in fact, a number of the important new drugs in the US approved by the FDA have been on much smaller population in the setting of relapsed refractory disease. That's how the IDH1 inhibitors got licensed. It's likely how the menin inhibitors will get licensed. And that's a little bit more like the position you're talking about. And thirdly, I think we should be forming alliances with patients to understand what is ethically acceptable and what the need is. That the whole world has changed. These questions weren't relevant 10 years ago because we didn't have the drugs. We now do have the drugs. You've generated them through your tax dollars, and you have every right to demand very easy access to them. So I think this is an area of creative discussion, but we should put the patient need at the heart of it, and we should be trying to get rid of regulation and bureaucracy if at all possible. In other words, I think I'm agreeing with you.